Here we go. Now we'll start the next phase of the chapter, which is how we actually capture the energy from the sunlight and turn it into sugar. So this is going to be happening only in plants, as far as we're going to define here in the chloroplast. There are other single-celled organisms that can do photosynthesis. If they're eukaryotes, they probably use the system that we're going to talk about, with chlorophyll A being at what we're going to call the reaction center. And then there are some bacteria who can fix energy from the sun and use it to generate energy and a source of food, but we won't focus on them. We'll come back to some of these discussions later on when we talk about how life evolved and how things changed on the planet. So we'll revisit that another time. Anyway, here we are in the chloroplast. So plant cell, cell walls. If it's a green cell, it probably has lots of chloroplasts in it. And we've talked about chloroplasts in the cell organelles chapter, so now we're just going to take it further and actually get into the chemical reactions that are taking place. There's two parts of photosynthesis. There's what they call the light reaction, and it's pretty obvious. This is the part that requires light, or a photon of energy, to activate electrons. We're going to move those electrons around, and I bet you can guess what we're going to do with electrons. We have electron carriers, we're probably going to have a chain. We're going to have oxidation reduction going on. And then we're going to use the energy generated. We're going to make some ATP. We're going to make a cousin of NADH. This is called NADPH. Handy that it's the one in photosynthesis. So you can always think about the NADP for photosynthesis, H. And then we're going to use those energy carriers, ATP and NADPH, to fuel what's called the Calvin cycle. This time they do preserve the name for the scientists who figured out most of those reactions. The Calvin cycle is a fairly complicated cycle that takes CO2 in, uses the energy to rearrange bonds, and make sugars. The first sugar that really gets taken out of the Calvin cycle will be G3P. It's one of the ones that was at the end or middle of glycolysis. We put that together to make glucose. We can make all different kinds of sugars that'll then get stored in the plant. So we need to talk about light for a little bit before we can move on to the light reaction and see how that energy is captured. So hopefully from your previous science classes, physics maybe, and definitely in chemistry, I know that you talked about how light is in the electromagnetic spectrum, and then visible light that we can see is only in a very tight, narrow range in that electromagnetic spectrum. Just to talk about the other parts first, we know that things that have very high energy, very small wavelength, are X-rays. We know that UV light has pretty high energy. We don't see it. The larger wavelengths with less energy in them are infrared and then microwaves. So let's go back to the area that we're going to focus on for the chloroplasts and what it can capture, and that's here, the visible light. Basically, we're going to see how there are pigment molecules inside the chloroplast that can capture these wavelengths of light. And interestingly, they do not capture the green wavelength. That's why the leaves look green. It's being reflected back into your eyes. So, what's happening with this energy? So a photon of energy, or light, comes in at a certain wavelength that has a certain amount of energy in it. And again, this is something that you did in chemistry. So remember, electrons can be elevated from a ground state to an excited state. And when you did labs with this last year, I know that you looked at an evacuated tube that had only one kind of gas in it maybe, and you watched it get energized with energy. The electrons got excited, and then they fell back down to their ground state and emitted that energy in the form of light, and you got to see a spectrum that those different compounds, or actually elements, emitted. So what we're going to do in photosynthesis is that light will be activating some electrons, and you'll see where they are in a minute. They get excited to another level, and then instead of letting that electron in an excited state just fall down and emit light, we're going to take that excited electron and move it down an electron transport chain, very similar to the one that we saw in the mitochondria. Use that transfer of energy to make ATP, and then do another process that also makes the NADPH. So how can we see that this is happening, and how can we figure out the wavelengths that it's doing it with? We can look at the pigments in a leaf, and we'll actually do this in the lab. We will squish out the pigments in a spinach leaf and separate it with a procedure, and then actually see which pigments can capture energy. We're going to see that, or we won't actually do this part of the experiment. We know that chlorophyll tends to absorb energy in this low range, in the blue and purple range, and it also absorbs energy in the orange to the red range with a peak in the red side. This is the 680 complex. We know that plants also are able to capture energy in the middle range here. Actually dips down a lot more in the green range that's not really showing up in this picture. And there are other pigments in the plant, particularly in the orange area, that help capture that energy for the plant. So what they've got mapped on here is, on the x-axis we have wavelength, and in the y-axis we either have absorbance of the light or an activity measurement. So how could you imagine, I'd like to think about this for class, that you could make an action spectrum, so you could detect whether photosynthesis is taking place. And a hint is here. They've got a very simple aquatic plant that's green, so it absorbs the energy from the sun and it does photosynthesis. And I'd like to think about a couple of ways that you could prove that photosynthesis is taking place. And that's basically what they're trying to plot in the blue line, that photosynthesis tends to peak at these particular wavelengths. If we would shine only certain wavelengths of light on this guy, we would then detect the highest amount of photosynthesis in these ranges. You do get a little bit of photosynthesis in this range, but it's much less. We'll focus on that as we move through the chapter and we actually do the labs. So what's capturing this energy? We'll break it down into two slides, but basically inside the chloroplast we know we have these discs or flattened membranes called thylakoid membranes. When they're stacked up on themselves, they're called a granum. When you have them in plural, they are grana. And those thylakoid membranes are where the chlorophyll molecule is embedded. So in that bilayer that makes up those thylakoid membranes, we have this thing called the reaction center, and it's filled with the different pigment molecules. There's a bunch that surround on the outside, embedded in this complex that has proteins, and then a very special one in the middle called chlorophyll A. And just to look at the structure of chlorophyll a little bit more carefully, you can see this area down here is a tail. That's a hydrocarbon tail. And remember, hydrocarbon tails are very hydrophobic. So this is the region that's embedded in the membrane. It's linked up with some polar molecules that helps keep it tethered in the membrane. And then we've got this huge thing called a porphyrin ring. And a porphyrin ring has these alternating double and single bonds. We've got some nitrogens in there. Remember, at every vertice there's a carbon. And then we coordinate a magnesium in the middle. This ring allows for the absorption of energy when the light hits it, and basically those electrons, because of resonance, just bounce around and get excited, and then eventually they will transfer that energy to a protein that will be next to it. The difference between chlorophyll A and chlorophyll B is just this little group up here. Chlorophyll A only has a methyl group, a CH3, and chlorophyll B has a aldehyde group, CHO. We're going to see the different colors that these guys have, meaning which they're absorbing different colors and reflecting different colors. They have a very slight color change. We'll do that in the lab. So this is what happens. This is the whole deal of the light reaction. We have, imagine that there's a membrane here, and that this reaction center is complexed in the membrane, like I showed you a minute ago. And light can hit this reaction center. Let's just focus on this side first. And light's bouncing around and finally gets transferred to the P680 complex here. Unfortunately, it's called photosystem 2. It's called photosystem 2 because it was discovered after photosystem 1. But after significant work, they know that this one goes first in the scheme of connected proteins that we'll talk about. So light hits it, moves around, finally activates this guy. When that happens, it ejects 2 electrons. So electrons move to a higher energy level, and then they get caught by proteins that are up here. Those proteins then get reduced because they gained electrons, and they send their energy down an electron transport chain that's not unlike the electron transport chain that we talked about in the mitochondria. It pumps hydrogens to the middle of the thylakoid space, and then we use ATP synthase to make ATP. But that's old hat now, so we don't have to talk much more about it. Then those electrons continue to move down, whereas in the mitochondria, oxygen was the last electron acceptor. In this case, it's the other photosystem, who simultaneously is being hit by photons of light, ejecting its electrons and needing a replacement. So electrons get ejected from P680 chlorophyll A, go up to a higher energy level, get captured by a protein acceptor, sent down an electron transport chain. That electron transport chain pumps hydrogens. Those electrons now move down in and replace the electrons that were ejected over here. So what happens to this guy? How does he get his electrons replaced? That's actually how we get the oxygen that we breathe. It happens right here in photosystem 2, or in the beginning of the light reaction. So water is hit by an enzyme that's in this complex, and 2 electrons are ripped out of that water molecule, and then we release a half of an O2, or an oxygen, and then 2 hydrogen ions. Those hydrogen ions are available to get pumped for this electron transport chain we just talked about. The oxygen finds another oxygen and diffuses away as the gas that we can breathe. So it wasn't until photosynthesis evolved at this level that we started to make oxygen in our atmosphere. Again, we're going to revisit that idea at another point in the class. So, electrons get ejected because the photons hit it, get back replaced from a water molecule, use up the water, split the O2, or secrete the O2. Electrons fall down the electron transport chain, you make ATP. Those electrons then fall into the P700 complex because this complex, this chlorophyll, is activated by 700 nanometers of light. Those get ejected. These electrons here can go down to another protein or another electron carrier which activates an enzyme that turns NADP into NADPH. So that's how we make our NADPH. This is how we make the ATP. So it turns out, things are never easy, right? It turns out that we actually need more ATP than we need NADPH in the Calvin cycle, which is what we're making this energy for. So over here, they neglected to keep in the P680 complex. He's running around doing what he likes to do. P700 has a protein up here that's very sensitive to the needs of the system. And sometimes it does not send those electrons over to make NADPH. And instead it goes and it sends it right back down that first electron transport chain. So I think having both of them there is better. They call this one, the first one we talked about, the Z scheme. Kind of looks like a Z or maybe an N. And then your electrons leave and finally reduce your NADPH. The other option would be that the protein up here that's detecting these electrons notices that it needs more ATP, and then basically, since this is all in the membrane, it can kind of float around. It goes this way and drops off those electron transport chain. And you see I'm making a circle here. This is called the cyclic flow of electrons. So in the light reaction, we can activate electrons to do the Z scheme and make both ATP and NADPH, or we could activate the electrons and do a cyclic flow to make more ATP. Bottom line is light hits the photosystems, you make ATP, make NADPH, which are going to go to the other part of the chloroplast, and you also split your water molecule into oxygen and hydrogen ions.